turn this on.
Can you hear me? Good. We hear a lot about data sets that have been generated by top-down, high-budget genomics. But a few years ago, we set out to develop methods by which an individual lab interested in a specific biological process could map the transcriptional network that regulates that process using an R01 scale budget. And to do that, we took a two-pronged approach. Along the first prong, we developed a novel method that we call net profit for mapping transcription factor networks using gene expression data. And we developed and validated that method using existing data from Saccharomyces cerevisiae. At the same time, we collaborated with the Tamara Deering lab to attack a practical problem in transcription factor network mapping, in which we had to generate the data as we went along in a sort of pay-as-you-go process. As I said, the method we developed is called net profit. I'm not going to say a lot about that today because it was published already last year, and it's the subject of my highlights talk on Tuesday if you want to hear more about it. But I will briefly give you the intuitive idea behind it, and that is it ranks all transcription factor target pairs by a score that's computed by combining a lasso regression coefficient between the transcription factor's expression level and the target's expression level with a log odds that the target is differentially expressed when the transcription factor is deleted. And this has some nice features. It's a purely expression-based method. And since expression profiling is itself a commodity method, this is scalable and relatively low cost. Surprisingly, we found using the S. cerevisiae data that this method was able to identify direct targets of transcription factors as accurately as the existing ChIP data for yeast. And finally, whereas ChIP gives you all binding events, this gives you only functional direct regulation. So the problem that we attacked involves this pathogenic fungus called Cryptococcus neoformans. And what we tried to do was to map the network by which Cryptococcus regulates the size of its polysaccharide capsule. Cryptococcus is an opportunistic pathogen that kills about 500,000 people a year, mostly untreated AIDS patients. And its lineage diverged from that of Cerevisiae not long after ours did. And so it's in principle quite a different beast. The important thing about the capsule is that it is small when the cells are living out in the environment, and it grows large upon infection of a mammalian host. And it has to grow large or the Cryptococcus can't maintain the infection. So the capsule is a critical virulence factor for Cryptococcus. One of the major challenges we faced in doing this project was figuring out which transcription factor deletion strains to make and profile. If I didn't say it already, the data that net profit requires is expression profiles from transcription factor deletion strains. So there are lots of choices. We wanted to pick ones that were relevant to capsule. We tried two approaches. The first approach we tried, which is described in a 2011 paper, was to prioritize for deletion transcription factors whose expression level is correlated with the size of the capsule across multiple capsule-inducing conditions. And then we later developed a method that is about to be published that we call phenoprofit. It's very simple. What it does is it takes the net profit predicted targets of a transcription factor and calculates how enriched they are for genes that are already known to be involved in the process of interest. We rank order transcription factors according to that enrichment, and then we go down the list and start deleting them. What we find is that a surprisingly large number of them turn out to be involved in the process of interest. So in all, the Deering lab made 41 transcription factor deletion mutants. The transcription factors were selected, some because they were already known to have altered capsule size, some because they were predicted by the correlational method, and some because they were identified by the phenoprofit method. As a result of that, we identified 17 transcription factors that were whose deletions had an altered capsule size, and that was not previously known. So here you see the images of the capsule for the 17 novels arrayed from smallest capsule to largest, with wild type somewhere in the middle. We wanted to evaluate how good the capsule size correlation method for prioritizing transcription factors was at identifying transcription factors that would have a capsule size phenotype as compared to the net profit method. And we also threw in the comparison a method that was published a few years ago by the Ed Marcotte group called phenologues. And the basic idea behind phenologues is that genes that work together to achieve some common function in one organism are likely to work together to achieve a common function in another organism, even if the functions themselves have diverged. So using that principle and knowing some capsule-involved transcription factors, we can identify other predicted capsule size involved transcription factors. We also threw in a really sort of simple-minded baseline method for comparison. That's based on the idea that we should prioritize transcription factors whose expression level is significantly different in capsule-inducing conditions than in non-inducing conditions. So using the 41 mutants that we made and their capsule size phenotypes, along with 10 other ones that had appeared in the literature, we carried out basically made an ROC curve for each of these 4 methods. And what you see is that essentially phenoprofit is the only one that works for this phenotype. So in summary, we developed a workflow in which we start with transcription factor deletion. We make transcription factor deletion strains. We use RNA-seq to create expression profiles. We use net profit to create network models. And from there's several different ways you can go. One is you can apply phenoprofit to the network models in order to prioritize more transcription factors for deletion and expression profiling, and then sort of go around the loop again. Alternatively, when you're satisfied with the network, you can kind of create network products from the predicted targets of each transcription factor. So example products are you can infer transcription factor binding motifs, and you can infer transcription factor functions by methods like Go enrichment analysis. We were pretty confident that net profit works well based on the findings using the Cerevisier data, but we wanted to spot check the Cryptococcus data, partly because we were using RNA-seq rather than microarrays. And so there was one existing published ChIP-ChIP data set for Cryptococcus, and that's for a transcription factor called GAT201. The Deering lab carried out ChIP-seq on 2 additional factors. Oh well, not important. That's okay. So the 2 additional factors are USV1 and NRG1. So we then took the top 40 net profit predicted targets of each of these transcription factors, and we calculated the enrichment of those 40 for ChIP-positive targets relative to what you would expect from 40 randomly selected genes. And we found that the top 40 for USV1 were about 4-fold enriched. The top 40 net profit targets for GAT201 were about 25-fold enriched. And the top 40 for NRG1 were also about 4-fold enriched. And then we did the same thing for the top 80, the top 120, top 160. And so you can see what the trends are. We also took the net profit predicted targets for each of these transcription factors and ran a motif inference program on the promoters of the predicted targets. We used FIRE, and we obtained the 3 motifs that are shown on the top of those alignments. And then we did the same thing for the ChIP-seq targets, and we obtained the 3 motifs that are shown below in the middle. And what you can see is that for USV1, the net profit motif and the ChIP-seq motif aligned very well. For GAT201, you also see a strong similarity between the two motifs. For NRG1, there are some significant divergences. So we took the best homologue that we could find in S. cerevisiae, for which there was a known transcription factor binding motif. We used Gary Stormo's SCRTF database and pulled out the best motifs for these. And what we found was that in the case of USV1, the Cryptococcus motif, which both the expression data and the ChIP data agreed on, had diverged from the cerevisiae motif. For GAT201, you find the expected GATA family resemblance for all three motifs. And for NRG1, the cerevisiae motif supports the ChIP-seq motif over the net profit motif. So net profit isn't perfect. Its targets, in this case of NRG1, we think are contaminated by some indirect targets that are probably direct targets of a transcription factor called MBS1, which is itself a target of NRG1. I'm just going to take a couple minutes to tell you about a few of the findings that we got from analyzing these data. One thing we wanted to do was to understand the transcriptional dynamics of capsule induction. So we did a time course of RNA-seq after transferring cells from non-inducing conditions to capsule-inducing conditions. And then we took all the capsule-involved TFs, and we clustered them into 4 groups based on their temporal expression pattern. So you can see that we then took those groups, and we looked for regulatory relationships between them that were enriched. What we found is that there were this revealed a hierarchy of regulation, in which group 1 sits at the top. It regulates groups 2 and 3, and both of those regulate group 4. We did the same thing with the non-transcription factor target genes that are known to be capsule involved, and those clusters are shown in the rectangles. And what we see is essentially in this analysis is a lot of coherent feed-forward loops. So for example, group 1, which is expressed in this inverted checkmark pattern, represses group 2, which is expressed in the checkmark pattern, and they both have opposite sign effects on the same groups of target genes. So this makes sense, and it gives an idea of causality about how these shapes, these temporal expression patterns, are coming about. We also laid out the biochemical pathway that makes some of the precursors, the activated sugar precursors for the polysaccharide capsule, and then mapped out the transcription factors that regulate each of those. And what you see on this is the genes that have a smaller capsule upon deletion are shown in blue, and the ones whose deletions create a larger capsule are shown in yellow. So one of the exciting things about this is that it allows you to understand why the transcription factors have the mutant phenotypes that they do. For example, the transcription factor SEER1, when you delete it, you get a small capsule. It is an activator of 4 different enzymes and transporters that are involved in making capsule precursors. When you delete any of those, you get a small capsule. And so you can understand why deleting SEER1 gives you a small capsule. Conversely, USV1 deletion has a hypercapsular phenotype. And you can see that it actually represses 3 different enzymes and transporters that themselves have hypocapsular phenotypes. So you see the expected sign change of phenotype for repressive edges. I could talk about this network all day, but I think that gives you a good feeling for some of the analyses that we've been able to do with it and some of the insights that we've had. So I want to summarize by saying that genomic discovery can be biologically focused. So it's possible to do genomics with a pull style rather than a push style of data generation. TF perturbation plus RNA-seq plus net profit together yield good direct functional networks. And phenoprofit is able to effectively identify TFs that are relevant to a biological process of interest. Only if you can do that can you actually generate the focused data set. Biologically driven network generation and analysis yield biological insights. And finally, I want to give you a little preview of where we're going with net profit. And that is that we'd like to take the expression data, use the existing net profit algorithm to generate networks, use the predicted targets of each transcription factor to do motif analysis, and then use those motifs to rescore the targets, hopefully driving down the scores of some of those that are not direct targets and bringing up the scores of some that are direct targets. So in conclusion, I'd like to thank my collaborator, Tamara Deering, and in her lab, Stacy Gish and Joe Wang, and graduate student in my lab, Zeke Mayer, who's about to finish up, and a former graduate student, Brian Haynes, who developed net profit. Thank you.
So, in a nutshell, what do you attribute the success of net profit to?
Good question. So I think it's the differential expression. I think that when you have differential expression data, so lots of people have developed algorithms based on regression across lots of data sets. But regression just is very weak compared to knocking out a transcription factor and seeing what changes. Now, you would expect that the things that change include some indirect targets, and that's true. But if you score them according to how reliably they change or how much they change, the ones at the top empirically are the direct targets. So we describe that by saying that the signal from deleting the transcription factor dissipates rapidly in the network.
Any other questions?